Yeah, hello everyone. Sorry I can't be there today. My name's Sophie Ward. I'm a postdoctoral researcher at the School of Ocean Sciences, Bangor University. I work closely with Pete Robbins and Stuart, but there have been other modellers on the Ecostructure project too, Matt Lewis, Alice Gower-Brown and Jonathan Demmer. As a team, we've been looking at modelling larval dispersal and gene flow in the Irish Sea on both work packages 4 and work package 8. The modelling that we've been doing is called biophysical modelling, sometimes referred to as larval dispersal modelling or particle tracking modelling. We have a number of tools and we've developed a number of tools over different projects at the School of Ocean Sciences with many different applications, but basically the fundamentals of biophysical modelling, they're based on a hydrodynamic model. So we have an ocean model of the Irish Sea, or many of them, and we can apply those at different spatial and temporal scales to depending on the question, the research question being asked. But we have applied within the Ecostructure project on questions such as understanding potential dispersal for marine non-native species, particularly in the nearshore region, and trying to understand marine population connectivity. We have applied, actually, that follows on nicely from Morag's talk. We're already applying some of the tools developed during the Ecostructure project to other projects. So there's a project we did with NRW and Zoological Society London, looking at modelling eDNA transport in the Irish Sea along the Welsh coast, and then also applying the models to another ERDF funded project called the Shellfish Centre. So we're a heavy user of supercomputing whales, so a little plug for the usefulness of that there. The Irish Sea itself is a really interesting place for us to model, to simulate the currents. So it's really energetic. We have deep energetic headlands. We have very quiescent shallow bays, all of them in short spatial scales. So often what we need are actually very high resolution models to resolve those currents and flows. Also within the Irish Sea, there's the extra complication of seasonal stratification, whereby in the summer months, as the surface waters warm up, we get a bit of a density front between the warmer waters shown up there on the top left figure. The warmer waters are indicated by the yellow, and then the cooler mixed waters that remain mixed throughout the season and the whole year. We get a density front between them, and that's really important actually for dispersal of material, because it can act as a barrier for dispersal, but also if larvae or matter become entrained within that frontal system, it can actually act as a conduit, and it can really influence long-term dispersal of materials. The first thing I looked at within the Ecostructure project is the importance of spatial scales on simulating the flow and the dispersal and transport of materials within the nearshore region, looking within the first couple of kilometres of the coastline. Often what we find in biophysical modelling is that because of hydrodynamic models available, we actually tend to use some coarse resolution models. So we wanted to better understand what this meant for what we're simulating and how well we're simulating it. There's a little video there showing a trajectory, so the little path followed by a hypothetical larvae in models of different resolutions. So I developed a few models of different spatial scales, 50 metre, which is the orange trajectory, up to a scale of 10 times that at 500 metres, which is the green trajectory. And what we found was that actually if we were using relatively low resolution models, they were overly retentive in the nearshore region, so they were not transporting material out from the first couple of kilometres offshore into the offshore, first couple of kilometres inshore. They weren't transporting enough material offshore. They were underestimating that offshore transport. Further still, they also underestimated the potential dispersal distance of the particles, again, because they were retaining material within the nearshore zone. And they also, because of everything retained within that nearshore zone, what they were doing was the low resolution models were overestimating a longshore connectivity, which can have implications when we're looking at trying to simulate connectivity in the nearshore region. This is an online tool that I know that Stuart's going to talk about later on, but I just wanted to plug it here because it's online, it's freely available to use. Alice Gower-Brown did the background hydrodynamic modelling and Jonathan Demmer was involved too. We've used this already as a prototype for another modelling tool, much like this, for looking at microplastic dispersal in Indonesia. So it was a brilliant idea and really well applied. And we had a webinar, and I'm sure Stuart will talk to you about that. Okay, some use of a biophysical model for looking at genetic connectivity in the Irish Sea. This was Melanie Prentice's work, what went alongside her work. And what we did was apply the ocean model, and it was an output used by the Met Office, so it was in collaboration with them. It was three-dimensional hydrodynamic and particle tracking modelling. And we looked at how well connected Melanie's various sites were depending on the hydrodynamic flows within the Irish Sea. Her points are in the left plot, and in the right plot, don't worry about the detail, but what these are connectivity matrices, and I wanted to put them in here because actually they're a really brilliant way for us to quantify and understand the number of connections between discrete sites and also the strength of those connections, and we can really interrogate what makes those change. So there are 4 connectivity matrices there, and the numbers within them are years to connected. So if they're yellow, it doesn't take very long for the sites to become connected. The point here being on the top left of those 4 grids is June, and then it goes June, July, August to September down to the bottom right. And you can see in September there are far fewer connectivities between those sites. And the point here to make is that that can really influence, so the timing of spawning, or the time within the water column that particles or larvae are within the season, can really affect their dispersal patterns. So as we see warming waters and changing hydrodynamics with climate change in the Irish Sea, it's likely that we're going to see changes to the background connectivity patterns. Staying on the theme of future things, this is going into work package 8 modelling. We have been looking at how the incorporation or the installation of various offshore renewable energy structures may affect the background connectivity within the Irish Sea, in the sense of using those offshore or species using those structures as what's known as marine stepping stones. So there's just a schematic there of sites A to B, which otherwise may not be connected. However, if larvae are able to foul on those structures, they may be able to do this stepping stone year by year spread through those offshore structures to site B. So that's setting up some artificial connectivity between those sites. What we also wanted to look at here was not only potential for marine stepping stone dispersal now, but how that might change with climate change and changes to hydrodynamics within the Irish Sea. On the left-hand plot there are some sites that we have released some particles from, and looking at the background connectivity between those sites. I'm sorry there's no coastline on there, but hopefully you all know the Irish Sea well enough to be able to cross your eyes and see that that's Wales, some of Ireland and England. And then the next step was looking at the connectivity between potential planned offshore renewable energy structures, which have been mapped there on the right. So those have been simulated too, and then we can combine the 2 connectivities and have a look at how those offshore renewable energy structures affect the year-on-year spread of particular or coastally spawned species. An example here again, just to quantify with these connectivity matrices, I've pulled out site 4 there, which is in the dashed little circle on the left-hand plot. And so everything that spawned from site 4 was theoretically connected there to sites 3, 4, 5 and 7, looking at the connectivity matrix on the right-hand side. And I've circled those points for you in the left-hand plot, but the point there being is that it's a really neat way that if we change the connectivity, we'll be able to do a comparison, a real quantitative comparison between how much incorporating those installations may affect the background connectivity, and we can then compare that with how much the changes due to climate change and warming seas may be affecting the connectivity too. And the way that the climate change may affect the connectivity is through warming of the waters, which also may affect the background hydrodynamics. Looking on the left-hand plot here, this is it's just an example plot of potential changes in monthly mean sea surface temperatures in the Irish Sea for 30-year chunks in the past. So that was 1990 to 2020, and then into the future, 2060 to 2090, and it was based on a scenario from the Met Office. Now, the Met Office have actually just made available to us some much more comprehensive climate change projection scenarios of the Irish Sea, which is really exciting, and that's now work ready to be done. And the idea here being that is with warming waters, what happens is that larvae have shorter pelagic larval duration. So the number of days or the amount of time that they stay within the water column until they mature and are able to settle out tends to decrease with warming waters. And that's a plot of that relationship. And I've taken that from the literature, and I haven't referenced it. So sorry, that's an O'Connor 2007 or 2014 paper. Stuart can correct me on the year on that. But what happens is, so I had a look in the Irish Sea and I did some comparisons, and we're looking at about a 20% reduction in pelagic larval duration based on those temperature differences into the future. So if a larvae now stayed in the water column for 10 days, now maybe come the end of the century they'd only be staying in the water for 8 days, and that can really affect the background dispersal and then the subsequent connectivity between populations. So to summarise, in this ocean modelling and the biophysical modelling, we've developed a number of tools. We're applying them to various research projects. These we're outputting these in the form of papers, publications, which we're keeping a broad audience in mind so that they are applicable to a wide ranging audience. There's ongoing project legacy. We're already applying the tools that we've developed, and also I've really enjoyed being on the project. I'm really sorry I can't be there today. It's been fun, so thanks for having me.